Welcome to G Fun Facts Online. Today we are diving deep into the biological reality of the human body, specifically dismantling the idea that our organs function in isolation. The concept of a heart that simply pumps and a kidney that simply filters is an anatomical convenience, not a biological truth. Nowhere is this distinction more blurred or more clinically critical than in the intimate and often deadly embrace between the heart and the kidney. This is the cardiorenal axis. For decades, medicine treated heart failure and kidney failure as separate entities, often managed by different specialists in different wings of the hospital. But the body knows no such boundaries. When the heart falters, the kidney suffers, and when the kidney fails, the heart breaks. This bidirectional relationship is known clinically as Cardiorenal Syndrome, or CRS, and it represents one of the most complex, lethal, and intellectually fascinating frontiers in modern medicine. To understand CRS, we have to look beyond the macroscopic mechanics of blood flow and urine output. It requires a descent into the microscopic and molecular world, a world of crosstalk where organs speak to one another through chemical messengers, genetic signals, and metabolic shifts. This is the story of that conversation. The realization that the heart and kidney are inextricably linked is not new. In 1836, the pioneering physician Robert Bright, famous for describing Bright's disease, observed that patients with advanced renal disease often possessed enlarged, structurally abnormal hearts. He was the first to posit that altered blood from the failing kidney could poison the heart. However, for nearly 2 centuries, this connection was viewed largely through a hydraulic lens. The heart was the pump, the kidney was the filter. If the pump failed, the filter dried up due to low perfusion. If the filter clogged, the pump got backed up with volume overload. This hemodynamic hypothesis dominated medical thought until the early 21st century. It was only in 2008, under the leadership of Claudio Ronco and the Acute Dialysis Quality Initiative, that the medical community formalized the definition of cardiorenal syndrome. They established a classification system based on 2 axes: primary organ dysfunction and acuity. We have type 1, or acute cardiorenal syndrome, where acute heart failure, like cardiogenic shock, leads to acute kidney injury. Type 2 is chronic cardiorenal syndrome, where chronic heart failure causes progressive chronic kidney disease. Type 3 is acute renocardiac syndrome, where acute kidney injury, perhaps from contrast nephropathy, causes acute cardiac dysfunction like arrhythmias. Type 4 is chronic renocardiac syndrome, where chronic kidney disease leads to cardiovascular disease, such as hypertrophy or atherosclerosis. Finally, type 5 is secondary, where systemic conditions like sepsis, diabetes, or amyloidosis simultaneously attack both organs. While this classification was a triumph of clinical taxonomy, it barely scratched the surface of the why. Why does a patient with stable heart failure suddenly develop kidney failure despite normal blood pressure? Why does a young dialysis patient have the calcified arteries of an 80-year-old? The answers lay in the molecular crosstalk. Before diving into molecular biology, we must acknowledge the physical forces at play. The hemodynamic interaction is often described as a balance of forward flow and backward pressure. First, we have forward failure, or low flow. When the heart cannot pump effectively, resulting in reduced cardiac output, the kidney perceives a threat. Specialized baroreceptors in the carotid sinus and efferent arterioles of the kidney sense this low-volume state. In response, the kidney aggressively retains salt and water to boost blood pressure. In a healthy person, this is life-saving. In a heart failure patient, it is disastrous, leading to fluid overload. Then there is backward failure, or congestion. Modern research has revealed that venous congestion is perhaps even more damaging than low flow. The kidney is encapsulated in a tight fibrous sheath. When the right side of the heart fails, pressure backs up into the inferior vena cava and eventually the renal veins. Because the kidney cannot expand, this increased venous pressure collapses the delicate microcirculation within the kidney, halting filtration. This concept has spurred the development of novel pusher devices to assist perfusion and puller devices to aid decongestion. However, hemodynamics alone cannot explain the full picture. Clinical trials have shown that even when flow is restored, kidney function often does not recover. This suggests that there is a molecular memory of damage, a biochemical conversation that continues long after the physical pressure is relieved. The crosstalk between heart and kidney is mediated by a vast network of neurohormonal and inflammatory pathways. When dysregulated, this homeostatic conversation turns into a symphony of destruction. The renin-angiotensin-aldosterone system, or RAAS, and the sympathetic nervous system are the first responders to organ stress. When renal blood flow drops, the kidney releases renin. This sets off a cascade, producing angiotensin II, a potent vasoconstrictor. Angiotensin II saves the immediate circulation but slowly destroys both organs. It induces cardiac hypertrophy, thickening the heart muscle, and renal fibrosis, scarring the kidney. It also stimulates aldosterone, which causes sodium retention and promotes collagen deposition in the heart and kidney, stiffening both organs. Simultaneously, we see sympathetic overdrive. The failing heart screams for help via the nervous system, triggering a massive release of epinephrine and norepinephrine. This fight-or-flight response becomes permanent. The constant bombardment of catecholamines is toxic to cardiomyocytes and induces constriction of renal blood vessels, starving the kidney of oxygen. A critical aspect of this is the phenomenon of sympathetic crashing in acute cardiorenal syndrome. In the setting of acute decompensated heart failure, the sudden surge in catecholamines can lead to acute kidney injury through rapid ischemia and intrarenal redistribution of blood flow away from the cortex, where the glomeruli are, to the medulla. This cortical steal phenomenon leaves the filtration units of the kidney hypoxic and vulnerable to necrosis. Beyond the nervous system, the body enters a state of sterile, chronic inflammation. This is not an infection, it is a metabolic fire. Venous congestion in the gut causes the intestinal wall to swell and leak, known as the leaky gut hypothesis. Bacterial toxins, primarily lipopolysaccharides, translocate into the bloodstream. The immune system recognizes these toxins and launches an attack. Macrophages release pro-inflammatory cytokines like tumor necrosis factor alpha and interleukin-6. These cytokines are cardiodepressants. They directly reduce the contractility of the heart. Simultaneously, they attack the kidney, causing tubular cell death. Recent attention has focused on the NLRP3 inflammasome, a protein complex inside cells that acts as a sensor for danger. In CRS, the NLRP3 inflammasome is perpetually activated, churning out IL-1 beta and IL-18, driving fibrosis in both organs. Surprisingly, the spleen has also emerged as a player in this axis. The cardiosplenic axis involves the release of immune cells from the spleen in response to cardiac stress. Following a myocardial infarction or acute heart failure event, the spleen discharges a reservoir of monocytes. These monocytes travel to the kidney, infiltrate the tissue, and differentiate into inflammatory macrophages, creating a second hit to the kidney. Then there is oxidative stress, the rusting of the axis. Every cell needs oxygen to produce energy, a process that creates byproducts called reactive oxygen species, or ROS. In health, antioxidants neutralize ROS. In CRS, this balance collapses. The endothelium normally produces nitric oxide to keep vessels relaxed and healthy. ROS destroys nitric oxide, leading to endothelial dysfunction. The blood vessels become stiff and constricted, increasing the workload on the failing heart and choking the kidney. Perhaps the most exciting recent discovery in cardiorenal medicine is that both heart and kidney failure are fundamentally metabolic diseases. The adult heart is a voracious consumer of energy, deriving up to 80% of its ATP from fatty acid oxidation. It is a high-efficiency engine. The kidney, specifically the proximal tubule, is also highly dependent on fatty acid oxidation. In cardiorenal syndrome, both organs run out of fuel. To survive the low oxygen environment, they revert to a primitive fetal metabolic state. They switch from burning efficient fats to burning inefficient glucose through glycolysis. While glycolysis requires less oxygen, it produces far less energy. The heart becomes energy-starved, unable to relax between beats, known as diastolic dysfunction. The kidney tubular cells, unable to maintain the massive energy gradient needed to filter blood, shut down or die. This switch is driven by the downregulation of PGC1-alpha, the master regulator of mitochondrial biogenesis. Furthermore, mitochondria are not static. They undergo fission and fusion. In CRS, this dance is disrupted. The protein DRP1 is upregulated, driving excessive fission. The mitochondria fragment into tiny, dysfunctional spheres that spew out ROS and trigger cell suicide. Concurrently, MITOFUSIN2 is downregulated. The ratio of MITOFUSIN2 to DRP1 is now considered a molecular thermometer of cardiorenal health. If inflammation is the fire, fibrosis is the scar that remains. Fibrosis is the final common pathway of almost all forms of CRS, replacing functional tissue with inert collagen scar tissue. GALECTIN3 has emerged as the conjunction junction of cardiorenal fibrosis. Secreted by macrophages, GALECTIN3 activates fibroblasts in both the heart and kidney, turning them into scar-building myofibroblasts. High levels of GALECTIN3 predict poor outcomes. Another pathway is the TGF-beta superhighway. Angiotensin II signaling triggers massive TGF-beta release, which is self-perpetuating. Once scarring starts, the mechanical stiffness stimulates even more TGF-beta release. But how does a failing heart tell the kidney to start scarring? The answer may lie in epigenetics and novel communication via extracellular vesicles. Cells release tiny bubbles called exosomes or extracellular vesicles. We used to think these were garbage bags, but we now know they are addressed mail packages. A stressed heart releases vesicles filled with specific proteins and genetic material. These travel through the blood, land in the kidney, and dump their cargo, effectively reprogramming the kidney cells to mimic the heart's pathology. Inside these vesicles are microRNAs, tiny strands of RNA that silence specific genes. For instance, MIR21 is the villain. It is upregulated in fibrosis, blocking anti-fibrotic pathways. Conversely, MIR29, the protector which normally suppresses collagen, is downregulated. This genetic crosstalk implies that we might one day treat CRS with infusions of antagomeres to neutralize bad microRNAs. We must also discuss the phenomenon of metabolic memory. Why do patients who have their blood sugar or blood pressure controlled still progress to organ failure? This is an epigenetic effect. DNA methylation and histone modification can lock cells into a pathological state. For example, a transient spike in blood sugar can induce specific histone methylation marks on the promoters of pro-inflammatory genes. Even when the sugar is gone, these marks remain, keeping the inflammatory genes primed. In CRS, studies have mapped aberrant DNA methylation patterns in the myocardium and renal tubule cells, explaining why early intervention is critical. We cannot talk about the heart and kidney without mentioning the gut, the third organ in this axis. As renal function declines, the kidney can no longer clear toxic metabolites. Simultaneously, gut edema alters the microbiome. Bacteria in the gut break down dietary amino acids into precursors that the liver turns into toxins, like indoxyl sulfate and p-cresyl sulfate. Additionally, TMAO, derived from choline found in red meat and eggs, is produced by gut bacteria. These toxins accumulate in the blood of CRS patients. They are cardiotoxins that stiffen the heart and induce arrhythmias, and they are nephrotoxins that damage renal tubules. Thus, the gut acts as a reservoir of poison that perpetuates the cycle of failure. Regarding diagnostics, for decades we relied on serum creatinine and ejection fraction. Both are flawed. Creatinine is a lagging indicator. By the time it rises, 50% of kidney function may be lost. We are moving toward structural biomarkers like NGAL, the troponin of the kidney, which spikes in urine or blood hours after injury. KIM1 is another marker for tubular injury. Soluble ST2 and GALECTIN3 offer prognosis regarding fibrosis and stress beyond standard testing. Finally, let us look at the therapeutic revolution. The last 5 years have witnessed a golden age in cardiorenal therapeutics. We have moved from simply managing hemodynamics with diuretics to targeting the molecular core. The game changers are SGLT2 inhibitors like empagliflozin and dapagliflozin. Designed for diabetes, they stunned the world by proving to be potent treatments for heart failure and kidney disease, even in non-diabetics. Their mechanism involves the tubuloglomerular feedback. The macula densa is a cluster of cells in the kidney that senses salt delivery. In heart failure, salt delivery to the distal kidney is often low. SGLT2 inhibitors block sodium reabsorption upstream, increasing sodium delivery to the macula densa. The macula densa senses this and releases adenosine. This adenosine constricts the afferent arteriole, reducing the hyperfiltration pressure that damages the kidney over time. Effectively, these drugs rest the glomerulus. They also shift metabolism back toward ketone bodies, a superfuel for the heart and kidney. Then we have GLP-1 receptor agonists. The FLOW trial showed that semaglutide significantly reduces kidney failure and cardiac death in CKD patients by reducing oxidative stress and inflammation. Additionally, non-steroidal MRAs like finerenone selectively block the mineralocorticoid receptor, preventing fibrosis without the dangerous potassium spike seen with older drugs. Looking to the future, precision medicine and AI will help us identify which subtype of CRS a patient has, whether it is driven by volume, fibrosis, or mitochondrial failure. Gene therapy vectors are being designed to replenish proteins like SERCA2A in the heart and Klotho, an anti-aging protein produced by the kidney. We are also seeing the integration of bioelectronic medicine, with implantable devices that stimulate the vagus nerve to activate anti-inflammatory pathways. The Cardiorenal Axis is a reminder of the biological unity of the human organism. It is a story of how a hemodynamic insult transforms into a molecular tragedy. As we peel back the layers, from the gross anatomy of congestion to the subcellular dance of mitochondrial fission, we find new hope. We are no longer just treating heart failure or kidney failure. We are treating the cardiorenal syndrome, aiming our therapies at the molecular crosstalk that binds these two vital organs together. The future of medicine lies in this space between, the intersection where the heart meets the kidney. Thanks for listening to G Fun Facts Online.